So I'm very excited today to have Scott Linderman speak in the statistics seminar. Scott is an assistant professor of statistics and computer science by courtesy at Stanford. He's also an institute scholar in the Wu Tsai Neurosciences Institute and a member of Stanford BioX and the Stanford AI Lab. He was previously a postdoc with Liam Podinsky and David Bly at Columbia, and he completed his PhD at Harvard, working with Ryan Adams and Leslie Valiant. And today, I guess with that, I'll let Scott take the floor.
All right, thanks so much, Allie, and thanks everybody for coming today. It's really an honor to be able to present this work. Someday, hopefully, we'll get a chance to get down to LA and to see people in person. Very much looking forward to being able to travel again. So today, I want to tell you about some recent work. This is one of the first projects from my lab at Stanford since starting, as I was just saying, starting in 2019. And it builds on a couple of things that I've long been interested in: point process models and latent variable models, and then thinking about how to apply those types of techniques in neuroscience to the analysis of high dimensional neural spike train data, which I'll tell you a little bit about. So let me make sure that I'm clicking on the right stuff. Yep. Okay. So, one of the reasons why it's exciting to be working at this intersection of neuroscience, statistics, computer science is that in the field of neuroscience over the past decade, there's been some exciting developments on the recording technology front. There are now techniques for doing brain-wide imaging of neural activity, so genetically modifying model organisms such that their neurons fluoresce in proportion to their activity, and then being able to record in, for example, worms or fish or mice or a whole host of model organisms, the activity of hundreds, thousands, even tens of thousands of neurons simultaneously. Pair that up with advances in machine learning and computer vision for tracking and quantifying animal behavior with high precision, and we have the ability to start recording neural activity from freely behaving animals performing complex tasks and getting rich measurement of both what's going on in their brains and what are they doing behaviorally. And of course, the ultimate goal of neuroscience is to try to tie these two things together, to be able to tell a coherent story that starts with sensory inputs bombarding our neural systems, computation in those circuits, ultimately producing behavioral outputs. Now, from a statistician's perspective, these data sets and these technologies are interesting also because they're a great source of rich, high-dimensional data. So as I was saying, the dimensionality of these neural recordings might range from hundreds to tens of thousands of neurons. And I just saw some recent work that was claiming 1 million neurons simultaneously recorded. Those neurons fire spikes, action potentials, and these data sets can record anywhere from, or contain anywhere from millions to billions of such spikes. Behavioral videos is coming in with high resolution, maybe multiple cameras, and we're talking hundreds of gigabytes an hour of raw data. So these data sets are high dimensional, but they're also interestingly complex. They span multiple subjects, so individual mice, for example, multiple brain regions, multiple trials, multiple condition types, and different genetic variants of model organisms as well. So a whole host of diversity in these data sets. They contain many covariates, behavioral videos, but also sensory stimuli. And often we know something else about the system as well. We know something about how neurons maybe are connected to one another. Or perhaps we have genetic sequencing data, which tells us about different types of neurons, different cell types. Finally, there's been a trend in neuroscience towards studying not just highly constrained and highly trained behaviors, like reaching from the center of a screen to a target in the periphery, but rather towards studying natural or so-called ethologically relevant behavior that requires fewer experimental constraints, is perhaps more reflective of what these animals do in the wild, but at the same time leads to much less structured data set, time series that aren't so nicely aligned to the onset of particular trials. So these present challenges to us as statisticians trying to work in this domain. And that's where my lab is trying to focus our effort. And I've roughly divided up how I think about what we do into a couple of different buckets here. So we spend a lot of time thinking about 4 statistical modeling problems. So I'll talk today about point processes. We also do a lot of work in state space and dynamical systems models. Think about hierarchical and multimodal Bayesian models, graphs and networks, deep generative models, et cetera. And then, of course, Thinking about modeling leads naturally to thinking about algorithms. So how do we actually go and fit these models and infer something about their sets of parameters? So in this side of things, I'm thinking about variational inference, about MCMC and augmentation schemes and things of that sort. But both of these modeling and algorithmic efforts are ultimately in the service of computational neuroscience. So trying to think about how to develop models and algorithms that will allow us to probe different types of latent structure in neural spike train data or in high dimensional behavioral data. Thinking about even just the basic questions of how to get signals of interest out of these raw data streams, such that we can start to ask questions about the underlying computations that these circuits are performing, decision making, action selection, for example. Now, obviously, I won't tell you about all these different aspects of our work today. Instead, I'm going to focus on just one particular project, which has to do with point process models for detecting sequences in high dimensional neural spike trains. And this is work that was led by my postdoc, Alex Williams, in collaboration with a really fantastic now graduate student, then undergrad, Anthony DeGlaris, and longtime collaborator, Yixin Wang from Columbia, now at Berkeley. So the outline for the talk today is first I'm going to start by motivating sequences in neural spike trains, why we care about them, and what types of methods are currently in use for trying to detect and extract them. Then I'll frame another way of approaching this problem that addresses some of the limitations of existing techniques. It's a point process model framed in continuous time, and in particular, it's a type of doubly stochastic point process model called a Niemann-Scott process. We'll present a new algorithm for performing posterior inference in this model that draws on recent connections to mixtures of finite mixture models to develop a collapsed Gibbs sampling algorithm. And then finally, I'll show you some example results of applying these methods both in simulated and in real data from Songbird HVC, Higher Vocal Center, and rodent hippocampus. And I also want to say, even though this is over Zoom, so we're not all in the same room, feel free to do the equivalent of just raising your hand or shouting out a question at any time if you have any questions or clarifications. Okay, so diving right in, why do we care about sequences? Well, sequential spiking patterns are a recurring finding both experimentally in neuroscience, but also one that has a long-standing theoretical motivation as well. So to give you one experimental example, if you look at neurons in the hippocampus of, say, a rat running on a wheel, you will find neurons that are encoding the position along that wheel, essentially the phase of this rotation. And these are so-called place cells. These are neurons that encode position in the outside world. And if you looked at this firing of this activity over time, as it's running around in this, essentially in this ring, you would see a cascade of neural activity encoding the trajectory through space as it's reflected in the activity of these neurons. Similar, another example of sequential activity in neural data comes from looking at the higher vocal center in songbirds. It's particularly this premotor area that's responsible for producing the songs that these songbirds sing. So on the top of this plot, we're showing a spectrogram, which shows frequency over time. And these different red blips correspond to times when this bird is singing a different element or a different syllable of its song. And of course, these songs are critical to their survival. These are how they find mates. If you look at neural activity in this premotor area, again, you see cascades of neural activity that are encoding both position in the song, but also position in each individual syllable. So how much time has elapsed from the start of a particular element of this songbird's song. And these are two examples of many. So on the experimental side, there's examples of sequential activity that's been implicated in motor production and motor activity during sleep, perhaps as an element of replaying experiences from our awake behavior, and during decision making. On the theoretical side, sequential activity has been postulated to be a critical component of various aspects of neural computation. For example, in learning and memory, in representing time that's elapsed, in the function of hippocampus, as we're seeing on the left here, but also as a mechanism for storing memories over time in recurrent neural networks. So how do we currently go about finding such sequences of neural activity? Well, existing methods are highly supervised. They involve first looking at the behavior of, say, the songbird spectrogram to try to pull out instances of repeated syllables. So syllables AAA, which all have characteristic blips in the spectrogram, B and B a different blip, C a different one with a different duration. And then going and finding, once we've segmented time according to this spectrogram clustering, looking back at the neural activity in this part of the brain and saying, are there corresponding patterns of neural activity that occur at the same time or perhaps slightly offset from the motor behavior that we use to guide us. So just as an example, one thing that you might find, and this is just the schema here, is cascades of neurons in HVC firing in sequential order for each instance of a songbird's syllable. And then this is a premotor area. Those spikes in that part of the brain might then go on to produce the actual muscle movements that give rise to the vocal song that we've recorded. So this is all well and good if you have access to really clean behavioral data. And in the adult bird, we do. However, if you look at a juvenile bird, a bird that's just learning to produce this song, there's much less of a clear, discrete structure to guide us. So here's a spectrogram of what's called a subsong with proto-syllables from work about, well, now over 10 years ago from Dmitry Aronov in the Phi Lab at that time. And so here it's not so clear how you go about clustering this into different syllables to then go and look for patterns of neural activity that might encode them. Here's an example of actual spikes from such an untrained bird. What we'd like to do is, even in this case where the behavior is not so clear cut, we'd like to be able to tell whether or not there are sequential patterns of neural activity underlying this or hidden in this neural time series of spikes. So just to be clear here, each dot in this plot here is the time at which a corresponding neuron on the y-axis fires an action potential. So these are these brief blips in the membrane potential or the voltage of a single cell, which carry information between pairs of neurons. So while it's not clear from this raw data, if I told you that here's a permutation of the neurons, same spikes, but just permuting the rows, well, all of a sudden, a pretty clear pattern of sequential activity starts to pop out. And indeed, you can see by eye that there are basically two different types of syllables or behaviors that are being, or cascade types that are being hidden in this data set here. We've gone ahead and color coded them as red and blue. So we see many instances of the blue cascade, not necessarily tied to the precise timing of the red cascade, but often correlated there as well. So, we're not the first people to go after this problem of trying to identify such sequential activity hidden in neural spike trains. Indeed, there's a long line of work doing this, and perhaps one of the most popular methods currently for tackling this problem is a method of convolutional matrix factorization, which has popularly been called seq-NMF in the neuroscience literature, presented by Makavicius et al. In 2019. The way this approach works is you start by assuming that that spike train is roughly an outer product or a low rank matrix that is an outer product of two different types of things. There are neural motif factors, which correspond to the different sequences of activity. And then there's temporal factors, which tell us about the time at which those motifs occur. Now, it's not just your regular old matrix factorization, it's a little bit different. The difference is that here, the way that you reconstruct the matrix from this pair of neural and temporal factors is that every time you see a non-zero entry in H, the temporal factors, you convolve that with a corresponding instance of this neural template, the red cascade. So hence, this is called a convolutional matrix factorization in contrast to just your regular old matrix factorization. And the hope is that this convolution of temporal factors and neural factors is a rough approximation to the raw data that's been collected from this neural circuit. But the raw data on the right and the matrix on the left are clearly not quite the same type. We've got spikes on the right, perhaps a point process. On the left, we've got this matrix. So what's typically done is first to divvy up time on the right-hand side by binning the number of spikes in each bin of, say, width delta t, maybe anywhere from 10 milliseconds to 100 milliseconds, et cetera. And then a further step that's taken in this approach, the seq-NMF approach from Makavicius et al., is to further take those spike counts and just blur them a little bit to account for slight jitter or perhaps perhaps model misspecification between the convolutional method on the right and the actual data, or convolutional method on the left and the actual data on the right. Now, this binning and smoothing is obviously somewhat undesirable. So why is that? Well, it discards the precise timing of these individual action potentials, which could be quite important for very fast behaviors like execution of syllables in the songbird's song. It also means that slight variations in these sequences can lead to poor estimates and reconstructions of the underlying convolutional matrix factorization parameters. From a statistical perspective, it's somewhat lacking in terms of a probabilistic model. We don't have an explicit noise model or any sort of prior distributions on parameters that we could bring to bear. And it's not obvious how to embed this into some higher order model, hierarchical model to capture sequence to sequence variability. And all these limitations conspire to produce these types of performance degradations that we see in the model's predictions as we vary or corrupt the data in different ways. So for example, this plot is taken from that Makavicius et al. Paper. As we vary the probability with which individual neurons participate in a sequence, the ability of the model to extract the underlying ground truth sequences and simulated data falls off. As we add or jitter the times of those individual spikes, again, performance degrades. As we warp the speed at which individual sequences are executed, performance degrades. So all of these, I think, can be traced back to some of the limitations of this convolutional matrix factorization formulation. And that's what we're going to try to address in this talk. The way we'll do so is by framing this or reframing this problem in terms of a continuous time point process model. So before just jumping to the model formulation, I want to spend just a bit of time building up some core insights about Poisson processes and point processes more generally that I think many of us are familiar with, but this is a good groundwork on which to build for this talk. So Poisson processes, perhaps the first stochastic processes that we encounter, generate sets of random spikes. These are going to be the building blocks for this model of neural spike trains. Poisson process, as we know, is governed by an intensity function. We'll call it lambda of t. And what that tells us is that in any interval of time, the number of events or spikes that we expect to find is a Poisson distributed random variable whose expectation is the integrated intensity over that interval. And one of the defining properties of a Poisson process is that non-overlapping intervals of time are independent. The number of spikes in T1 is independent of the number of spikes in T2 if T1 and T2 are non-intersecting. Now, Poisson processes are nice for a number of reasons. One of them is that we have really great tools for thinking about how to simulate Poisson processes. So one way that you can simulate a Poisson process using that intuition we just developed, or that characterization from the previous slide, you could say, well, first, let me sample the number of spikes that occur in a total interval. That's just, again, a Poisson random variable integrating over the entire time interval that we are considering. And then the independence of disjoint intervals implies that we can sample the precise times of those spikes independently by drawing from a probability distribution whose density is the intensity, but normalized by the total measure, the total intensity integrated over that interval. Another nice feature of Poisson processes is the so-called superposition principle, which is just as in the sum of Poissons is a Poisson, so is the superposition of Poisson processes yet another Poisson process. So suppose that we have spikes that are union of spikes from k different independent Poisson processes. Their union is yet again another Poisson process, and the intensity of that Poisson process is just the sum of intensities for each individual component here in indexed k equals 1 to big K. So in pictures, it's what we see on the right-hand side. Suppose that we had a bunch of different things contributing to the overall intensity. Each one is a little Gaussian bump. We can sum up all of those Gaussian bumps to get a total intensity for this overall Poisson process. And again, we're just building up some fun facts about Poisson processes that we'll leverage when building up a model for sequences of neural spiking activity. But Poisson processes alone aren't going to get us there. And in particular, this assumption of independence is clearly one that is limiting for our purposes. If we want to have a cascade of spiking activity, then almost by definition, we need that spikes in some bin of time at time t must depend on whether or not there was a spike in that same sequence immediately preceding. So the way around this limitation is to build out this model one layer further. And that's what a doubly stochastic Poisson process does. Here we're going to model the intensity function as itself being a stochastic process. And there's a whole variety of doubly stochastic Poisson processes that have been explored in the literature that can give rise to different types of dependency structure. So we can have doubly stochastic processes that cluster spikes together or that repel spikes such that they're not too close to one another. And these go under various names, log Gaussian Cox processes or Matern, Strauss, Thomas processes. And a really nice book by Moeller and Wag-Peterson from 2003, I think characterizes the taxonomy of point processes very nicely. The particular type of doubly stochastic point process that we're going to use is what's called a Neyman-Scott process. And this is building on this paper from 1958, where they were interested in modeling the distribution of locations of galaxies in astronomical surveys. Now, of course, nowadays, astronomical surveys are much, much larger scale than what you see in this little snippet here, but the picture is pretty much the same. We want to find places in the night sky where there are perhaps clusters of galaxies all found closer together than you might expect by chance under, say, a homogeneous Poisson process. So how does this NSP work? Well, it's easy to introduce this via an example. So the way that you would sample a Neyman-Scott process is by first taking it in two steps. The process begins with a set of latent events, which we'll assume are just drawn from a homogeneous Poisson process over some, here shown as a 2D plane. And these locations of these latent events become the centroids of clusters of observed events. So what we actually observe are these orange dots, and they're realizations of a Poisson process whose intensity function is taken by summing up impulses centered on each one of these latent events. A lot like a Gaussian mixture model, that's sort of the intuition that you should have in mind here. So NSPs are doubly stochastic processes. Let's explain them a little bit more formally. As I said in words, we'll sample these latent events from a Poisson process. Let's call the set of latent events Z, indexed by K. Each latent event adds a non-negative impulse response to the intensity of our observed events. So we'll say lambda of T is a sum of impulses G. Each impulse may be centered on the location of the latent event Z. And then what we observed is a sample from this Poisson process whose total intensity is lambda of T. Applications go beyond just simply in astronomy. They also have been used in ecology as models for how forests grow over time, with trees following or dropping seeds and giving rise to new generations of trees subsequently. And as we'll see, they're pretty nice building blocks for modeling neural data as well. All right, now, here I've just said in step 3, sample from a Poisson process with rate lambda. Here's where we can go back and leverage that Poisson superposition. Lambda is just a sum of non-negative impulse responses. We can sample the set of observed events instead by first sampling each cluster independently from its simple Poisson process with just here a little circular disk representing its intensity. And then the union of those spikes drawn from each individual cluster is a draw from the Poisson process that we actually want. How do we sample each component? Well, again, we go back to our characterization for Poisson processes. We sample the number of spikes that occurs in each one of these clusters, and then we distribute their locations independently by sampling from the normalized impulse response. Okay, that was all shown in 2D. How do we adapt this type of model for modeling neural spike trains? Well, in particular for modeling sequences, that's what we want to build up to. Well, here, conceptually, we can think about this model from the same perspective, except here our latent events are a bit more complicated. So our latent events will consist of 3 things. They'll consist of a timestamp, here shown as tau. They'll consist of a type, which I've indexed, or which I've called R, and that's going to take on a finite number of different values. Think like red or blue. And then they'll consist of an amplitude, which tells us something about how many spikes we expect to find for a particular sequence. We'll set the background rate to not just be the simple homogeneous Poisson process. It'll be homogeneous in time, but we'll have a categorical distribution for the type and a gamma distribution or density for the amplitude. Once we've sampled these, it's the same as the Neiman-Scott process from before. Each latent event is going to add a non-negative impulse response. Here, our impulse responses will be modeled as Gaussian densities. They'll be centered on the time of the latent event, perhaps with a type-specific offset, which is redundant for the simple one neuron model that I'm showing here, but we'll make use of it in just a second. And then, as I said, the amplitude A sub k associated with this latent event is going to scale up the height of this impulse response to produce more or fewer spikes for any individual instance of a latent event. And then this lambda naught, this is our background intensity. Maybe not all of the observed spikes are going to be attributed to sequences. Perhaps there's other stuff going on in this neuron that's producing action potentials that are not directly the consequence of some underlying sequence. This is one neuron. To finish the story, let's adapt this to multiple neurons. I realized I was one click behind. So here's the realization at the bottom of this entire process, the superposition of spikes from each of these underlying latent events of types red and blue. Now let's finish this story up by explaining how to take this single neuron point process model and extend it to multiple neurons. So now we'll treat each neuron as a realization of a point process, but they all share the same set of underlying latent events shown up top. So now each one of these neurons gets a scaled version of or has an impulse response for each type. We've introduced just a few parameters. We've introduced an extra neuron-specific and type-specific scaling, because maybe not all neurons participate in the blue sequence. We've introduced neuron-specific and type-specific offsets, B, which tell us that neuron N might participate in the red sequence at some particular lag, and C captures the width or the standard deviation of that impulse response. And then what we actually observe is this marked point process consisting of both the times T sub S and the neurons associated with those spikes N sub S. That's the model. I want to pause here and just ask if there's any questions so far on the framing of this model before diving into the algorithmic component of how we actually are going to fit this.
So this model appears similar to latent Dirichlet analysis and topic modeling. Can you use the techniques that they use for topic modeling in this problem, or?
Yeah, it's very similar to LDA, and in a sense, I guess you could think about LDA versus this Neiman-Scott process. They both share this underlying sort of clustering of neurons. So maybe if you think of each sequence as a document, or each time bin, if you were working in discrete time, as a superposition of spikes that come from, say, a sequence and spikes that come from the background, then that analogy, I think, starts to really be borne out. The algorithms we'll use will have a similar flavor in the sense that we'll be assigning each individual spike to one of the underlying components of its intensity function, either to the background part of the intensity or to one of the underlying sequences. And then this collapsed Gibbs sampling algorithm, which we'll discuss next, is going to look a lot like the collapsed Gibbs sampling algorithms for LDA. So I guess the high level symmetry here is that both LDA and these Neiman-Scott processes can in some sense be thought of as additive Poisson models. And so the same types of techniques of thinning and attributing spikes to clusters or words to topics kind of make sense in both cases. That's a great connection.
In the previous slide, is K a random number? Is capital K known a priori?
That is not known. That is a random variable, and it's random in the same sense that a draw from a Poisson process could create anywhere from 0 to, well, the support of the number of spikes is 0 to to infinity. So the number k will actually be distributed according to a Poisson distribution because it's the number of spikes that comes from this underlying Poisson process. So that's actually a great segue into the next part of this talk, because that's what we're going to use to make a connection between the NSP we just developed and mixtures of finite mixture models, a type of non-parametric Bayesian mixture model. So
also one more question. So when you do that, they're color coded different spikes. In your example, you have only like 2 colors. So is that a number you want to estimate as well, or what's
right,
the physical meaning of this color here?
Right. So let me explain that one more time here. So the red and blue colors correspond to different types of sequences. So in the one neuron case, it's a little bit it's hard to make that distinction. When you have multiple neurons, think about the blue cluster or the blue sequence type as evoking neural activity on the first half of these neurons, the first 4 neurons. And that's what we see. Can you see my mouse actually? Maybe not.
We can.
You cannot. Okay. Well, what I'm trying to point at is, the first 4 neurons, the first 4 rows of this time series. Those are the neurons that fire spikes generally in this blue sequence type. The red sequence type is another type of sequence that produces spikes, but on a different set of neurons, maybe the latter 4 neurons in this picture. So that number of different types, blue and red here, that we're going to treat as a hyperparameter. Now, we're not going to be Bayesian about it or treat it as a random variable. We're just going to fit models with 2 types of sequences, 3 types of sequences, 4, and so on, and then ask which number of sequence types gives us the best fit according to, say, cross-validated log likelihood.
Okay, so is that what you call the motifs in the matrix factorization corresponding to the number of motifs?
Yes, that's right. So
okay, thank you.
Types, we're sticking with the same color scheme here. That's right. All right, thank you for the clarifying questions. So as I was saying before, there's a close connection to a variety of different latent variable models. Perhaps the closest are non-parametric mixture models, and in particular, mixtures of finite mixtures. So let me make that connection a little bit more precisely. First, what's the goal of this section? Well, we'd like to be able to infer a posterior distribution on the latent events, so their times, their types, their amplitudes, and also the parameters of this model, which include the per neuron offsets and amplitudes and widths and the background intensities, things like that. Now, as I was just saying, there are going to be some hyperparameters as well, the number of types, for example, the red and blue sequence types from before being one of them. So the observation, which is clear by now, is that these models are a lot like mixture models. So a longstanding and a clear idea toward approaching this problem is to say, well, let's try to infer the underlying mixture component assignment for each spike. Call these the parent variables, omega sub s, which are numbers 1 through big K, where K is the number of latent events that we're working with. So given the parents and the latent events and the parameters become easy to sample. So essentially having these parent variables makes our sampling problem a lot easier. The challenge, as was just brought up, is that the number of latent events K is not a priori known or fixed. So how has this problem been approached? Well, one type of approach is reversible jump MCMC. This is what's suggested in that book from 2003 that I referenced before. But these reversible jumps can be pretty slow, especially when what you're trying to do in each jump is propose to introduce a new latent event at a particular time, at a particular location with a particular amplitude. As these models get more complex, these proposals are being made in higher and higher dimensional spaces, and making intelligent proposals can be somewhat of a challenge and can lead to slower mixing Markov chain Monte Carlo algorithms. So the observation that is going to lead us to a different type of algorithm, a collapsed Gibbs sampling algorithm, is to say, NSPs aren't just any non-parametric mixture models. In particular, they're mixtures of what are called mixtures of finite mixture models, or MFMs. What differs from other non-parametric models here is that the number of latent events K is drawn from a parametric distribution. Here we know that it's a Poisson distributed random variable. Its expectation is the integrated measure of that underlying background or latent event intensity. There's been some exciting work over the past few years from Jeff Miller and Matthew Harrison developing a family of collapsed Gibbs sampling algorithms for certain cases of mixtures of finite mixture models, based on some connections they made to Dirichlet process mixture models, for which, of course, we have a whole host of algorithmic tools. Now, the connection is close, but it's not quite perfect. NSPs don't quite fit this framework because we have this background rate that doesn't generally appear in the MFM. And also the number of observed spikes depends on the number of latent events. If we have more latent events, we're going to have more induced impulse responses, more observed spikes in expectation. But nevertheless, we can still build on this work from Miller and Harrison. And what we do is we derive a prior probability distribution on the partitions of spikes under this Neiman-Scott process. This is the key step toward working with partitions of spikes rather than with parent assignments and explicit numbers of clusters. So I won't go through the details of this proposition, but suffice it to say that, with a bit of math, that's also, again, it parallels the approach taken by Miller and Harrison. We can derive this prior distribution on partitions. It depends on hyperparameters alpha and beta of that gamma intensity. And in particular, that gamma measure on the amplitudes of each sequence, the expected number of spikes that each sequence produces, is a really key assumption to making this work out in closed form. Now, once we have this prior distribution on partitions, we can condition on particular observed data sets and a particular set of spike times and neuron assignments, and we can marginalize out all of the other variables. So we can marginalize out the time, the type, and the amplitude of each individual latent event, and instead just deal with conditional distributions on the partitions of spikes directly. So here, P of X sub K is the marginal likelihood of seeing a collection of spikes, say the red spikes or the blue cluster of spikes, given that they all come from the same cluster, integrating out the time, the type, the amplitude of that cluster. Now, when we only consider moving around one spike's partition assignment at a time, a lot of terms are going to cancel such that the conditional distribution of the partitions takes on a very simple form. So the resulting algorithm, which will be a type of collapsed Gibbs sampling algorithm, or a collapsed Gibbs sampling algorithm, will move individual spikes around, assigning them to the background component with some simple probability that depends on the background rate and the amplitude hyperparameters, to an existing component that depends on the predictive likelihood of seeing that spike given the other spikes in that component, but also depending on the current size of that component and again, the hyperparameters of the amplitude, or to a new component, which probability depends on the marginal probability of a single spike cluster, and then this ratio of a function called V, which is defined back here in that proposition. It's some infinite sum of Poissons that Miller and Harrison say, this sum converges, and you can compute it out to sort of arbitrary accuracy using just doing it in advance because it only depends on the hyperparameters. So let's contrast this collapsed Gibbs sampling algorithm for this Neiman-Scott process to other collapsed Gibbs sampling algorithms for other non-parametric Bayesian models. Well, first of all, as we said, the model that we're working with has this extra background buffer, essentially a cluster that always is present and that has just a constant intensity over the entire interval. So that doesn't exist in your standard mixture of finite mixture model or your standard Dirichlet process mixture model. But the other components, the probability of adding a spike to an existing component or a new component, those do have analogs in both the MFMs and the DPMMs. And in particular, the form of these probabilities of assigning to an existing component are identical between the NSP and the MFM. And they're very, very close to what we see in a Dirichlet process mixture model, except for this additive term alpha, which is from that gamma prior on the amplitudes for each sequence. The Neiman-Scott process and the mixture of finite mixture models also have the same probabilities of adding spikes to new components, this ratio of the V's, which stands in contrast to a Dirichlet process mixture model, where that probability of creating a new component is just constant, gamma. So it seems like NSPs and MFMs are basically identical except for this background component. But here's an interesting observation. The new cluster probability in the NSP, if we plug in that big form for this ratio V, we'll see that again, a lot of terms cancel. And this turns out to yet again be a constant, just like in the Dirichlet process mixture model, which is perhaps a little bit surprising. It suggests that there's an even closer relationship between Neiman-Scott processes and Dirichlet process mixture models than what we at first observed. Now, the specific relationship is that we can recover a Dirichlet process mixture model as we take a particular limit. As we take the limit that alpha, the concentration of our gamma prior on amplitudes of sequences, i.e. the expected number of spikes attributed to each sequence, as that concentration goes to 0 and the background rate goes to infinity such that the product goes to a constant, then we get back out the Dirichlet process mixture model updates from before. Now, intuitively, we can think about this because the DPMM, or think about this as saying that the DPMM is the limit of having infinitely many latent events, each of which has amplitude going to 0. And in hindsight, this relationship makes a lot of sense because in this limit, that random intensity function that we're getting under this Neiman-Scott process is an instance of a gamma process, which once normalized gives us the Dirichlet process that underlies DPMMs. So I think this is a really interesting takeaway. It positions these NSPs at midway point between sort of general mixture of finite mixture models and our classic well-studied Dirichlet process mixture models. And we get a knob, or rather two knobs, we get alpha and mu naught, which we can vary to sort of pick out points that are intermediate in this continuum of non-parametric mixture models. All right, that was derivations. Now, of course, we also implemented this, and Anthony, the undergrad, now graduate student at Stanford that I mentioned before, implemented it in Julia, and you can see it on our lab webpage. And this code is remarkably fast. So we can run thousands of Gibbs sampling iterations on hour-long recordings with neuropixel probes that contain millions of spikes. Each update of this collapsed Gibbs sampler goes through each spike, attributes it to one of the existing sequences or to the background. And in action, it looks like this. This may not render all that well on Zoom, but what we're seeing here is over the course of Gibbs iterations, this code is finding these different spike assignments. It's inferring the parameters of each individual sequence type, i.e., which neurons participate in it, at what lags, and with what variance. And then after each sample, we're permuting the rows to try to highlight or pull out this separation of spikes into two different groups of neurons participating in two different types of sequences. So I highly encourage you to check out the code. It's I think, very well written and very usable as well. So let me show you some results of applying this in simulation and in real data. So as I said, we tried this out first in synthetic data just to make sure everything worked as expected. And what we find well, first of all, just a little side point that in order to assess these models, you have to be a little bit careful about how you hold out data. The way we're going to do this, first of all, is by holding out data, measuring the likelihood that the model assigns to that held out data based on the inferred latent events or sequences given the observed spikes. But you have to be a little careful about how you hold these out such that you can accurately make these types of predictions. So you want to see, basically, at each point in time, you want to see something about which spikes are present. for each neuron, you want to see something about its spiking activity at least some points in time. And so that suggests this sort of speckled holdout pattern shown in the bottom left panel. So we sampled data with 2 different sequence types, multiple instances of both the red type and the blue type, plus a lot of background spikes on top of it. And then we held out some instances of time, like I was just saying before, and we try to predict or impute the times of these latent spikes. We can look at log likelihood on the train and the test set, and we can use this to guide this choice of the hyperparameter for the number of sequence types R. And we find here that indeed 2 sequence types maximizes this cross-validated likelihood. So this was just, again, sanity checking, make sure that everything works. We also then went and looked at this in more realistic data sets here of recordings made in the rat hippocampus by the Buzaki lab, where the rat is running down and back along a linear corridor. And so what you should see are spikes that fire as the rat runs down the corridor in one direction, and then another set of spikes that occur as the rat runs the other direction back. And that's because these particular cells don't just encode the position, but also the direction in which the rat is traveling. So here, this is, well, each scale bar is about 25 seconds. So we're looking at just a couple minute snippet from a couple of dozen neurons here. And we used this as, again, another proving ground for checking how well this model works, or illustrating how you could do hyperparameter selection in this model to see which parameters the model is most sensitive to. So here we see, again, a pretty nice rise in held out likelihood as we go to 2 sequence types, maybe enough evidence to suggest that there's 3 or 4 as well, but most of the gains are made going up to 2. We don't see a lot of influence of warping, which I'll talk more about in a second. And we do see, this is really just to show that you can use this type of technique to set all of these other hyperparameters as well. In terms of runtime, so the sampler appears to converge and faster than real time. So if we have an hour-long recording on just a simple laptop, this thing seems to plateau in terms of both training and both log joint probability, but also other metrics of Gibbs sampler convergence. And for very long recordings, we can parallelize the Gibbs sampler by taking well-separated chunks of time and just estimating or inferring their latent parameters or partition of spikes independently in parallel, because with very, very exceedingly low probability will they share the same underlying set of sequences. I started by making the case that the existing methods which work by discretizing time and doing some smoothing are insufficient. So we went back and we tested that quantitatively. And we found that we tried to reproduce this panel from the Macavicius paper, but in our hands. And for some reason, we transposed A and B in our NeurIPS paper. So I've transposed them back the other direction so that they match the panels on the left. What we find in all cases, as we vary the probability with which neurons participate in a sequence, or the extra amount of background spikes, or the jitter with which which neurons fire spikes, or the speed at which sequences unfold, in all of these cases, this point process formulation is much more robust to these types of variations than the convolutional matrix factorization methods. And here we measured that by looking at area under the ROC curve for trying to detect when there was, or how many different spikes there were, spike sequence types there were, et cetera, and which sequence they were assigned to. Now, we don't just get out point estimates and predictions of sequence assignments. We get out a full posterior distribution from this Gibbs sampler. So here what I'm doing is contrasting the PP-seq approach, which is what we call this point process model, which involves finding and clustering spike times directly, to the convolutional NMF approach of binning, smoothing, fitting the convolutional matrix factorization, and then resorting. And what we find in our posterior distributions are, first of all, if we run independent Markov chains and compare the samples of number of sequences that we find, they're pretty consistent across independent runs of these chains, again, suggesting that these algorithms are converging reasonably well, or at least to the same region of this latent variable and parameter posterior. And we get probability of which two spikes are assigned to the same sequence, the same cluster. We also get posterior distributions on the parameters of the model, the amplitudes of these different sequences, or the offsets, et cetera. So again, sort of a nice way of quantifying uncertainty in these parameters. I've alluded to warping a couple of times now. So let me tell you a bit about how we did that, because it's a nice example of how we can take this baseline model and extend it in different ways. So here's an example where synthetic data, different sequences, instances of, say, the blue sequence can unfold very quickly or very slowly. If we throw the existing techniques, convolutional matrix factorization approaches at this, they really struggle in cases where there's a lot of variability in the speed with which these sequences unfold. However, if we extend ppseq to instead give, instead of just having a time and type and an amplitude for the latent events, we also give it a warping factor, where the warping factor scales up the offsets and the widths of these responses, these impulse responses. Well, then this model naturally can handle sequences that happen at different speeds, because each sequence instance gets its own latent speed associated with it. And there we do a much better job of, again, pulling out these sequence types, even in the presence of this time warping. So, looking ahead, what do we want to do with this? Well, I mentioned hippocampal replay. So finding sequences of spiking activity in the hippocampus while rats are asleep that may be reflective of, say, some experience that the rat had while it was awake and moving around its environment. So this paper from Davidson et al. 2009 is talking about how those replay events can happen 15 to 20 times faster than they would happen in real time. So you're not just reliving your experience at unit time speed, you're replaying it maybe 15 or 20 times faster during sleep, perhaps. So in order to better study these phenomena, we need ways to look at activity collected during sleep and to detect these sequences, and even though they're happening much faster than they would happen while awake. So this tool seems like a particularly well-suited tool for that job. So we're starting to make progress on applying these methods to that type of data as well. So in conclusion, I presented this method that we call ppseq. It's a type of Niemann-Scott process model, a point process for detecting sequences in neural spike train data. It works on raw spikes, no binning, no smoothing. We can find rapid sequences with low spike counts. It's fully probabilistic. It's a Bayesian model. You can express prior distributions on all of these parameters. And in doing so, we find that having this probabilistic model makes it more robust to different types of noise. It connects, I think, in a really interesting way, these classes of doubly stochastic point processes to non-parametric mixture models, MFMs and Dirichlet process mixture models. And in doing so, we can generalize collapsed Gibbs sampling algorithms for these other models to work with these doubly stochastic point processes. And then finally, I showed one example of extending this to time warping, but of course, there's a whole bunch of other types of properties that you might like to imbue this model with. Maybe sequences progress through, but they halt partway. Or maybe these spikes don't just have spike times and neuron labels. Maybe there's more that we know about individual spikes. Of course, the spike time comes often from some electrophysiological recording consisting of voltage traces over time. So can we build that information in? And while I've presented this model in the context of neuroscience, you can imagine how other types of domains might benefit from a similar approach. So one example that we've worked with also with the Bly Lab is in applying these to streams of documents that come in continuous time, each document consisting of some vector of words and perhaps being sent by a particular sender. So all of these point process models, I think, serve a much bigger role in the broader statistics and machine learning communities. I need to acknowledge Alex again, and Anthony and Dishin, who did a lot of this awesome work. I'm very fortunate to have a group of amazing students at Stanford. All of this code is online. You can see, this slide needs to be updated. You can see the preprint, or you can see the final version from NeurIPS. And I'd like to thank my funders and end with this very nice animation that we made to illustrate what we do in taking in spike trains, writing a lot of code, and hopefully gaining some insight from statistical models that we fit. So thank you very much again.
That was a great talk. Thanks, Scott. Do people have any questions?
I have
Yeah,
one question.
Oh,
Sorry.
okay, go ahead, Jane.
Okay, thank you. I wonder if for the neuron data, do you expect that there would be like physical location information attached to the neurons? Let's say if they all come from the brain, if you know which part of the brain, like their relative location, right, among these neurons, I imagine that could be very useful in terms of getting those motif patterns, right?
That's a really great point. Yes. So I didn't talk about it here, but the way that those spike trains get in or come into our hands is that somebody sticks electrodes into some part of the brain in some generally model organism, and different types of electrodes will give you different amounts of information about the exact location or relative location of the neurons that you record from. So the HVC songbird spikes that we talked about, those came from some tetrode array. And I think the same is true of the rat hippocampus data that we looked at here. But one of the exciting technologies that's being used much more frequently nowadays are these very high density silicone probes, where they are silicon probes, which are going through multiple layers of cortex and down into subcortical brain regions. And you can record not just, the times of action potentials, but also which electrodes along the length of that probe were recording that neuron. So you get depth information as well as the precise timing of spikes. And so you're absolutely right. Maybe these sequences are not only evolving over time, but also according to some sort of spatial propagation of spiking activity. It's not a thing we looked at, but that would be a great example of extra information that you get about spikes and sequences.
Oh, thank you. Thank you very much.
My question's kind of similar. I'm wondering if you've thought about looking at Hawkes models instead of Niemann-Scott models, which Hawkes models might let you better take advantage of the time, the fact that time moves in one direction. And so if there's some latent triggering that happens, then the points that are triggered should happen after it. And that would be true for a Hawkes model, but not necessarily true for Niemann-Scott.
Yeah, that's a really great question, and also one that's near and dear to my heart, because a big component of my PhD thesis was on Hawkes process models too. So yeah, I love that proposal. So we have looked at that a little bit. So the trade-off here, I guess, is if you think about the Niemann-Scott process as the equivalent of, say, like a latent variable model, then a Hawkes process is kind of the analog of an autoregressive model. And in these sequences, I think you're absolutely right that we could model them well with also an autoregressive process where spikes on one neuron then produce impulse responses on downstream neurons, producing these cascades. Now, one thing that's I think a little tricky here is that those same neurons can also produce spikes that are not part of cascades as well. And maybe, well, one of the things we're working on currently that I think might get to this question is, imagine that you had a Hawkes process where the impulse responses weren't deterministic, but rather stochastic, such that spikes may or may not induce an impulse response on downstream neurons, kind of depending on whether or not there's a sequence unfolding at that point in time. So yeah, again, I think it's spot on. There is definitely, I think, interesting work to be done building or combining ideas from NSPs and from Hawkes processes.
Thanks. Very nice talk.
Thank you.
Are there any more questions?
I do have a question. So if the parameters of the model are not known a priori, can you also fit them when applying this algorithm?
Yeah, so indeed, we do estimate a posterior distribution on many of the parameters in the model. The parameters that we are getting a posterior distribution on are the parameters that tell us about, with what offset does a particular neuron tend to fire in the context of a blue sequence? And with what variance does that neuron tend to fire in the context of a blue sequence? What probability does it fire a spike in the context of a blue sequence? So these are all things that we are estimating a posterior distribution on, also background rates and things like that. And they all turn out to have very nice, simple conditional distributions once you know the partition of spikes. Things we're not currently being Bayesian about, we're not putting any sort of prior distribution on the number of sequence types, blue and red. We're just fitting or doing posterior inference for models with 2 types of sequences, 3 types, 4 types, and then doing some model comparison using held out validation data. So I think the answer to your question is, in general, yeah, we are trying to get posterior distributions over all of these different types of parameters, or as many as we feasibly can. And I think we have a pretty good solution for how to estimate the parameters that we don't so easily know how to get a full posterior on.
Thank you.
I have a question. Have you thought about how to deal with instances where there aren't explicitly like 2 or 3 sequences, but more so like, let's say there are 3 motifs and 2 of them sometimes combine to work together for something? Have you kind of thought about that or looked at instances of that?
Yeah, so no, we haven't really, but I think that, what that might end up looking like is having some sort of, dependencies between the latent events themselves.
Mm-hmm.
And I think that makes the inference problem a bit more challenging. Certainly, when we derived the collapsed Gibbs sampling algorithm, we did use the fact that the number of latent events was, that the latent events came from a homogeneous Poisson process, which, of course, doesn't have these dependencies.
Mm-hmm.
But I think it's an interesting direction for future work. Just even as a simpler example, one thing that we would like to have is that you don't see 2 instances of the same sequence at almost exactly the same time. That would be evidence that we're like over segmenting the spike train data more so than this should be. And one way you might try to get around that is by having some sort of repelling point process prior on the latent events. But again, that adds a layer of complexity that we haven't really sunk our teeth into yet.
Cool, thank you.
I guess there are no more questions now. That was an amazing talk, Scott. Thank you so much for coming. It was really appreciated it. Hopefully you can come in person when things get a little more normal. It would be great to see you.
All right. Well, thanks so much again for the invitation and for all the kind words and the attention, the great questions. So take care, and I'll see everybody someday, hopefully soon.
Okay, thanks. Bye.
Bye.